Our report Forensic Chemistry and Toxicology. Part 1 Forensic Chemistry. Introduced to Forensic Chemistry and Toxicology. Chemistry is a branch of science that deals with the identification of substance of which matter is composed. Also sa Forensic Chemistry, the branch of chemistry which deals with the application of chemicals principles in the solution of problems that arise a connection with the administration of justice. The use of chemistry to support civil and criminal litigation. Blood examination. It has been called the circulating tissues of the body, carry oxygen, nutrients to all parts of the body, and carries carbon dioxide and otherwise products back to the lungs, kidneys, and livers for disposal. For every 600 red blood cells, there are about 40 platelets and one white cells. Elements of blood, 45% from elements or solid materials constituting chiefly and cells, namely A. Red blood cells, the most abundant cells in our blood. They are produced in the bone marrow and contain a protein cells, hemoglobin that carries oxygen to our cells, non-nucleus. Noted, hemoglobin picks up and drop off oxygen. They are part of immune system and destroyed infections. Elements of blood, 45% from elements or solid materials constituting chiefly and cells, namely A. Red blood cells, the most abundant cells in our blood. They are produced in the bone marrow and contain a protein cells, hemoglobin that carries oxygen to our cells, non-nucleus. Noted, hemoglobin picks up and drop off oxygen. B. White blood cells. They are part of immune system and destroyed infectious agents called with pathogens with nucleons. Plasma 55%. This is the yellowish liquid portion of the blood that contains electrolytes, nutrients, and vitamins, hormones, clotting factors, and proteins such as antibodies to fighting infection. Serum, a straw yellow colored liquid from the clotted blood is allowed to stand for some time and that clot contracts. Platelets, the clotting factors that carry in the plasma. They are clotted together in the process called clottation to seal a wound and prevent loss of blood. Possible combination of blood types. Sa phenotype, meron siyang A, yung mga single letter or A tapos B, tapos AB or O. Sa genotype naman, merong AA, AB, BB, BO, AO, and OO. Importance of identification of blood group data, determination, paternity and maternity. Determination whether a child born from married woman could have been fathered by a legal spouse. Determination whether a child could or could not belong to that given set of parents in the case of accidental interchange of infants in hospitals. Types of blood stain. Passive blood stain patterns created from the force of gravity. Single drop blood has fallen vertically, whether it be from an injured person or other object to land into another surface. Seminal fluid, a viscid distinguished fluid of the male reproductive tract consisting of spermatozoa, suspended as secretion of accessory glands, biological components of semen. Seminal fluid has characteristic alkaline odor o isa viscid gelatinous and sticky. So number 2, spermatozoa of sperm cells, or tinatawag, may Greek word to na, yung Greek word, sperma, which means seeds. Sa Latin word niya ito, meron siyang, sa Latin word niya tinatawag na something sown. Where to look for seminal stain, clothes, body, and crime scenes, examination of semens, and seminal stain, physical examination, microscopic examination of the sperm stain, and view under high power microscope, and the specific test for semen. Ultraviolet light examination, seminal stain, exhibit bright bluish fluorescence. Number 3, semens with dry, even stiff, stretchy feeling to a cloth, and produce slight dipping of colors with the disappearance of the odor. Number 4, have appearance or outline of contour map. Fiber analysis. Hair, especially of grooved skin, which occur everywhere of the human body except a palm of the hand and sole of the foot. It grows rate of 0.3 to 0.5 millimeter per day. Hair range from scalp hair, pubis hair, axillary hair. Preservation of semen. Number 1, place in the pillbox of the test tube, properly folded, sealed, and labeled. 2 kinds of hair. Merong real hair, yung mga general long and stiff. Tapos sa mga fuzz hair, merong mga generally short. Parts of the hair: root, stop, and tip. Portion embedded in the skin. Portion above the surface of the skins. Most dito yung mga destructive part of the skin. Tips, sometimes turn point to distal end at cut hair stop. Parts of the stop: cuticle, contracts. Medulla versus animal hair. Sa medulla hair na human, meron siyang smaller na index, tapos less than 1/3 yung yung size. Also sa animals medulla is very thick siya dito. Manipis siya, tapos 1/2 or greater yung size ng hair sa animal. Glass is a super cold liquid with possess high viscosity and rigidity. Composition of glass: silica, boric oxide, tapos and phosphorus pentoxide. Composition of glass: silica, boric oxide, tapos and phosphorus pentoxide. Types of glass fracture: merong radial fracture and concentric fractures. Radial fracture, primary fracture, assemble of spokes of a wheel where predating rod originates at a common point. Sa concentric fracture, secondary fracture, having a appearance circle around the point of impact, connecting one predating crack to the other, thus forming triangular piece of glasses.
Hi, I'm Andre Kayanan from Firm 4C, and I'm reporting the collection and preservation of drug evidence. Collection and preservation of drug evidence, collecting samples of dangerous drugs from subjects requires careful handling to maintain the integrity of the evidence. Here's the general procedure for collecting and preserving drug samples. First is collection. A is law enforcement officer or forensic investigator collect sample from subject person immediate surroundings. B, samples may include pills, powder, liquids, plants materials such as cannabis, syringe, or other paraphernalia suspected to contain drugs. C is if collecting samples from the subjects, proper protocols for search and seizures must be followed to ensure legality and chain of custody. Second is packaging. Each example should be packaged separately in a clean, dry, and sterilized container such as sealable plastic bag or vial. B. Proper labeling with details such as date, time, location, and the subject names or identification number is essential for documentation and chain of custody purposes. Third is preservation. A sample should be stored in a secure and controlled environment to prevent contamination, degradation, or tampering. B. Temperature and humidity should be controlled to minimize any changes in the samples over time. C. For liquid samples, ensure tight sealing to prevent evaporation or leakage. D. If immediate analysis is not possible, samples should be stored at an appropriate temperature to maintain stability until analysis can be conducted. Fourth is transportation. A sample must be transported securely and safely to the forensic laboratory or testing facility. B. Law enforcement agencies often have specific procedures for transporting evidence to maintain chain of custody and integrity. Fifth is documentation. First is detailed documentation of the collection process, including photographs if applicable, should be recorded to provide a clear record of evidence chain of custody. Second is any relevant information provided by the subject or observed by the collecting officer should be documented for context and reference during analysis. Six is analysis. First is once received at the forensic laboratory, samples undergo analysis using appropriate techniques such as screening tests, chromatography, or spectrophotometry to identify the presence of dangerous drugs. Second is preservation methods ensure the sample remains stable and suitable for analysis, providing accurate results to support legal proceedings. Next is collection and preservation of fiber as evidence. Collecting and preserving fiber as evidence in forensic investigation requires careful attention to detail and proper handling to maintain their integrity. Here's a guide on how to collect and preserve fibers effectively. First is documentation. Before collecting fibers throughoutly, document the location, context, and appearance of the fibers using photographs, sketches, and written descriptions. Note any potential sources of contamination or disturbance. Second is personal protective equipment or PPE. Wear appropriate PPE such as gloves and masks to prevent contamination of the fibers and protect yourself from potential hazards. Third is collection tools. Use clean sterilized tools like forceps, tweezers, or sticky tape to collect fibers. Avoid using fingers to prevent transfer of oils and other contaminations. Fourth is sampling. Collect fibers from relevant surfaces or objects using the chosen collection method. Carefully lift fibers from the surface without bending or damaging them. Fifth is packaging. Place each fiber in a clean, dry container or envelope to prevent cross-contamination and preserve the integrity of the evidence and avoid folding or crumpling the fibers. Sixth is labeling. Label each container with detailed information, including the date, time, location, and description of the collected fibers. Use unique identifiers to ensure proper tracking and chain of custody. 7 is sealing. Seal the container securely with evidence tape or seal to prevent tampering and maintain the integrity of the evidence. 8 is storage. Store the packaged fibers in a cool, dry environment away from the direct sunlight to prevent degradation. Avoid exposure to moisture, heat, or chemicals that could damage the fiber. 9 is transportation. Handle the packaged fiber with care during transportation to the forensic laboratory. Use secure packaging so to prevent shifting or damage during transit. Last is documentation. Maintain detailed records of the collection and preservation process. Include any handling or storage conditions. Document any changes or observation made during transportation or storage. Next is collection and preservation of tool marks as evidence. Collecting and preserving tool marks as evidence is crucial in forensic investigation to establish links between suspects, tools, and crime scenes. Here's the detailed overview of the process. First is scene examination. Forensic investigators carefully examine the crime scene to identify and document tool marks left behind by perpetrators. This may include marks on doors, windows, safes, vehicles, or any other surface that have been tampered with. Photographs are taken to capture the location, size, and orientation of the marks in situations. 2. Documentation. Detailed notes are made regarding the characteristics of the tool marks, such as their size, shape, depth, and any unique features. Information about the substance materials, for example, wood, metal, then plastics, ganon, is also documented as it can influence the analysis and interpretation of the marks. Third is collection of evidence. Once identified tool marks evidence is carefully collected using appropriate techniques to preserve its integrity. This may involve first is photography. High-resolution photographs are taken of the tool marks from multiple angles and under in different lighting conditions. Scale markers are included in the photos to provide the reference for size. Next is lifting. In cases where tool marks are found on non-porous surfaces such as a glass or metal, forensic tape or lifting devices are used to lift the marks from the surface. These lifted impressions are then transferred onto suitable backing materials such as lifting cards or gel lifters for preservations and analysis. Next is swabbing. If there is possibility of trace evidence such as DNA or residue being presented on the tool marks, swabs moistened with appropriate collection solutions such as sterile water or alcohol are used to sample the marks for further analysis. Next is collection of substances. In cases where the entire substrate bearing tool marks needs to be preserved, such as damaged door or window frame, the entire item may be collected and transported to the forensic laboratory for future examination. Fourth is preservation. Proper preservation techniques are essential to prevent contamination, degradation, or loss of tool marks evidence during storage and transportation. Preservation methods include first is packaging. Tool mark evidence including lifted impression swabs and substrate sample is packaged separately in clean sterilized containers or envelopes to prevent cross contamination. Each item is labeled with relevant information including the date, time, location, and case number. Next is sealing evidence. Sealing. Evidence containers are securely sealed and initiated by the investigating officer to maintain the chain of custody and ensure the integrity of the evidence. Next is storage. Tollmark evidence is stored in a secure climate-controlled environment at the appropriate temperature and humidity levels to prevent deterioration. Access to the evidence is restricted to authorized personnel to prevent tampering or loss. Lastly, is transportation. Evidence is transported to the forensic laboratory following established protocols and chain of custody procedures. Special care is taken to prevent damage or alteration during transit, and evidence handling protocols are followed to maintain its integrity.